The next important technique is microdissection. The word dissection means to cut into pieces. So the dissection with the help of microscope, or we can say that the type of dissection in which the microscope is used to assist the dissection is called microdissection. Usually microdissection is used to remove tumor granules from the cell tissue, for example like from the brain, heart or nerve cells, etc. In the technique microdissection, the image is seen on a large TV. A variety of technique is used in the microdissection. The two main techniques are chromosomal microdissection and laser microdissection. In chromosomal microdissection, as the name indicates, glass needle is used to remove the portion of the diseased complete chromosome. In laser microdissection, it is also called LMD, laser light or beam is used to remove a single cell or entire tissue. Such type of laser microdissection is usually used in cancer research, in neurology, etc. Laser microdissection is one of the contact and contamination free method. That's why it is widely used today in medical field.